Steve Okonek, and Mike Bertram. So the webinar series is sponsored by the University of Wisconsin Division of Extension and the UW Nutrient Pest Management Program. I'm Jerry Clark. I'll be your host today, and I'm the Crops and Soils Educator serving the Chippewa County area. While many of you are familiar with the Zoom technology we'll be using today, just a few housekeeping things to go through. If you can keep your microphones muted, that helps with the extra noise, and the videos off also helps with some of the streaming issues that some of us might have out in parts of the state. Also, feel free to type questions for our presenters into the chat box. We'll have some time for questions at the end of the presentations. That chat box is located at the bottom of your screen if you need to find that, and we'll try to answer those questions, as many as we can, throughout the program. We are offering a CCA credit today, or 1.5 today, for our webinar. There will be a link with the form at the end of the program today. You'll be able to scan that code, or at least put that number, your certification number, into the form or the chat box, however, and we will get that to the American Society of Agronomy, so you get credit. If you have any questions or difficulties today, Jordan Schuler is our Zoom technology wrangler today. Jordan will put her information in the chat box, and if you have any connection issues, you can feel free to contact Jordan via email. With that, I'm going to stop sharing this stuff, and I will introduce our first speaker. That will be Dr. Mark Renz. Many of you know Mark. He's our weed science specialist and professor with the University of Wisconsin-Madison. He's going to discuss what are herbicides reducing the establishment success of your alfalfa. So Mark, we'll give it a try here and see if the technology holds up for us.
Yeah, great. Thank you very much. Can you hear me, Jerry? And
yep, you're
Mike, can you see my slides?
I can see your slide. So far, so good. Looks good.
Okay, great. Well, it's a pleasure to be here and talk to everyone. Hopefully we're all getting ready in the southern part for this big snowstorm that who knows if it will appear or not. So glad you're here today to talk about forages. And what I'm going to talk to you about is the use of herbicides, and are herbicides impacting the establishment success of alfalfa? We've had a lot of struggles with alfalfa establishment, and there's a lot of concern that herbicide carryover is a main issue. So that's what I'm going to talk to you today about. But first, I want to just kind of step back and talk about alfalfa. You know, in production systems, there's a lot of problems going on. We're having issues with soil erosion, water runoff, nutrient leaching and movement off site. We've got herbicide resistant weeds and just difficult to control weeds that are just really problematic. And I want to just emphasize that alfalfa is a solution to many of these problems. When we incorporate alfalfa into the rotation and alfalfa is growing, we get, large reductions in soil erosion and water runoff. This is just some data from some of our study plots. Nutrient leaching and movement off site is dramatically reduced with alfalfa. We've done lots of work with alfalfa and controlling water hemp, and we find that if we manage that alfalfa correctly, the water hemp is controlled without any additional inputs. It doesn't impact forage quality, and we almost eliminate seed production. And the same is held true for giant ragweed when we're growing alfalfa. So I want to emphasize that alfalfa is a solution to many of these problems we have. And so if you don't have it, you have these problems in some of your fields and you don't have alfalfa in there, you may want to consider adding that into the rotation. Okay, I'll get off my soapbox a little bit, but I'll say the challenge is in order to gain this success that I talked about, we have to get good, robust, well-established fields, like you can see in these pictures. And we've had some challenges in the past years in getting good establishment of our alfalfa fields. There's lots of reasons we can have poor establishment success, like you can see in this picture. A lot of site-specific issues can happen. I think in many cases, we're not following proper agronomic practices. We're not getting good seed to soil contact. We're planting at the wrong time. Our fertility or our management isn't correct. We can get weather issues and sometimes pests, phantomycetes, insect pressure can cause some of those issues. However, there's an additional potential stress and reason for successful alfalfa or for poor alfalfa establishment. And I think I'd like to take this quote from one of our national agronomists who recently has stated that over 50% of his fields that he's visited that were failures and he evaluated, he determined that over 50% of the reason those fields failed was the herbicide carryover. So clearly he's seen this as a new issue and is really problematic. So what I want to do today with the limited time is just explain what herbicide carryover is, if you're not familiar. Why does it appear to be a bigger problem than it has in the past? And then just kind of summarize what you can do. There's not a lot you can do, but there are some things you can do. So realize that when we apply some of our herbicides, they persist in the soil after application. And as many of you know, that's a benefit, right? We want that. We want those herbicides to persist and control those weeds that are going to emerge later on in the crop, either as a and now with herbicide resistant weeds like water hemp that emerge throughout the season. We're even applying these pre-emergent or residual herbicides with our post applications, and they really have some good benefit for weed control. However, in some cases, those residual herbicides can persist after we harvest that crop. And if they don't degrade to low enough levels, they can cause injury to the subsequent crop. So this is a great graph from Penn State that you can see. I'm going to move my little mouse here. So up here, we have large amounts of herbicide in the crop. And in this case, we're saying corn is the crop. And it's killing the weeds, but when it drops below a certain present concentration, it may have poor weed control. But realize it still has to drop in concentration to a very, very low level in order not to injure alfalfa. And so that's just a depiction that shows that. Give me a second to get my. So labels always provide some really good guidance on these plant back restrictions so that we don't have that. It's required by law to do that. But unfortunately, sometimes the wording is confusing, and it also can become difficult. You may have intended to apply it at a certain date, but it had bad weather or it was too windy, so you applied it at another date. So it's easy to forget this timing when you're planning on your next crop. So I just want to show you some examples of some labels. I'm trying to go across companies just to highlight some of the challenges. So first, I want to show Atrex. This is atrazine, and you can see the highlighted number 6. It says, do not plant sugar beets, tobacco, vegetables, or spring-seeded small grains or small-seeded legumes and grasses the year following application, or injury may occur. So that's clearly saying you have to wait a year or you could get injuries. Here's where it can get more confusing. Here's Corvus. We look on the Corvus label and you look down to alfalfa, which is in the lower left-hand corner. You see the rotation interval is 17 months. But now we're seeing all these footnotes. And so you really got to read the footnotes. So that's, it needs 30 inches of cumulative precipitation to plant to the next crop. And then let's look at that footnote number 3. When the soil pH is 7.5 or above, we're going to change that interval from 17 months to 24 months. So again, reading a lot of those footnotes can really, really change what that plant back restriction is. And I want to emphasize that. And I want to spend a little bit of time on Resicor because it's being widely used throughout the Wisconsin and the upper Midwest and has great control. But this really highlights how reading the fine details is really important. So if we look at the table on the Resicor label, we see alfalfa has a 10.5 month rotational interval. So if we're using it as a pre-emergence, it shouldn't be a problem if we're planting alfalfa the next year. But we can see there's a lot of these footnotes. Footnote number 2 applies to Idaho, Nevada, Oregon, Utah, and Washington, so we can ignore that. But we can see next to the 10.5 month, a 7 and an 8. So we got to go down and read those footnotes. And what I want to focus on is footnote number 7, where it says if Resicor is applied after June 1st, rotating to crops other than corn or grain sorghum the next year may result in crop injury. Okay, so again, we need to read those footnotes to make sure we're within those label restrictions. So we have these restrictions on these labels and they're complicated, but why are we all of a sudden now over the next couple of years seeing increase in herbicide carryover in establishing fields of alfalfa? Well, I think, you know, one of the big reasons is that we're using more residual herbicides in our crops that are rotated with alfalfa. And really what I'm focusing on is corn, but it could be soybeans or wheat. And these tend to have longer plant back restrictions. And to make matters worse, where we historically have planted those as pre-emergence when we're planting these crops, now because of resistant weeds like water hemp that emerge throughout the season, we're applying these post-emergence later on in the summer for herbicide-resistant weed management. It does a great job, but it increases our risk and our ability to miss that window may happen. So I want to just show you, I summarized some data that Rodrigo, my colleague, who is a weed scientist in corn and soybeans, has the common corn herbicides in Wisconsin. And what I did is just summarize, I think this is around 20 or 25 of these residual corn herbicides that we use. About half of them have less than a 10-month plant back restriction to alfalfa, which is the blue bar. But 40% of them are between this 10 and 12-month time frame. And so I want to emphasize that it's really easy to make a mistake, or not a mistake, but make these applications and plant alfalfa and not be in compliance depending on when you use these products, especially if you're using them post-emergence as a mix in with your post products. And then we have 10% that have a longer than 12-month restriction. So we have a lot, half of our products are 10 months or greater that are common corn herbicides. So we need to remember that. We also need to consider weather can really change what these, the carryover potential. In drought years, we tend to get more persistence, and we can talk about that if people have more questions as why. And then we've been having a lot of precipitation during those post-emergent applications. So therefore we delay them 1 or 2 weeks. And that delay of 1 or 2 weeks may be the reason we're seeing more of these issues. What I'm not going to talk about, but there is some data with some products that the frequency that we apply these herbicides may increase their persistence. And that's a very herbicide specific issue. But that may be if we're using a lot more of this herbicide repeatedly in our cropping system, it may be the reason why we're seeing more issues. Okay, let's get down to the nitty gritty of this. So what are the active ingredients we're concerned about? In wheat, I pulled out 4 products that are 10-month or more plant back restrictions to alfalfa. And you can see the ones that I would really worry about would be mesotrione, flurancelam, fluroxapyr, and clopyralid. In soybeans, surprisingly, even though soybeans are a legume, we have 13 products we need to worry about. I think really products that contain sulfentrazone, chlorimuron, or fomesafen are the ones we really need to worry about there. But it's really the corn products that we are really concerned about. Forage Genetics has a really nice list of a whole bunch of them that they're worried about, and they've created some nice fact sheets. There's over 15 products that have 10-month or more restriction. And there's lots that I've listed here of active ingredients that I'm concerned about. We've really kind of focused on clopyralid, that active ingredient, which can be used as a standalone in several of our products, such as Stinger, or is mixed with several of our products in corn, such as Resicor is one of them. And alfalfa is very susceptible to clopyralid. So we've been doing a lot of work in bioassays, trying to really understand that level of susceptibility. And I just want to show you some of the results that we've seen from some of these initial studies and talk about some of our future studies. So what we're doing with these assays is we're taking soil, and we're taking actually highly organic matter that has over 10% organic matter soil, potting soil, and we're mixing known concentrations of clopyralid in the soil. In this one experiment, we went from 10 parts per billion up to over 50,000 parts per billion. And we chose that because the literature says 10 parts per billion is really at the level where we start to see injury. Maybe a little bit higher that, and 50,000 parts per billion is the maximum allowable amount of clopyralid that can be in forage, grass forage that's fed to animals. So we figured that was a good range. And we grew we set up those concentrations, we planted alfalfa seeds in them and grew them for 21 days and then harvested the above ground biomass. And as you can see in this picture, you can see that there's a very clear curve where we're getting reductions in biomass as we're increasing those densities. Clearly at 400 parts per billion, you can see less than 100 and 100. You can see less than 40. And I just have cut off 10 because I was trying to just show the big pictures. When we put this in a graph, it gets pretty concerning. Here's the graph that we have here. We can see that there's, and this is on a log scale, so keep that in mind, but we see a linear response on a log scale, whereas as we're increasing those herbicide concentrations, we're getting less and less above ground alfalfa biomass. And so what we can learn from this was just a few pots in this initial study, is that very, very small amounts of clopyralid in the soil can reduce alfalfa biomass. And we can see that, in the hundreds of parts per million for sure. The big question is, how does this relate to the levels when we're spraying? We're not spraying in parts per billion. We're spraying on weight per acre. And so some of the future studies that looking at comparing those results to other results. We've just started another study that I just want to say that we're better trying to understand how this relates to application rates. So stay tuned for some of that information. And oh, by the way, soybeans are really sensitive to clopyralid too. So want to just point out, even if you don't like alfalfa or aren't growing it, you need to be aware of this with soybeans as well. We saw at 400 parts per billion, a significant drop in soybean biomass after 21 days. So I guess the question I would just raise is, is this a problem in our fields? I think when we're not following the label rates and the plant back restrictions, we're clearly seeing issues and we're getting reports of those issues. But I wonder out loud is, do we really fully understand the impact these herbicide residuals have on alfalfa establishment? All of these studies that are conducted and these studies that are conducted to support registration, 99% of them are done as greenhouse trials. They're short run times, 21 to 8 days like we showed, where I wonder, and they're evaluating above ground biomass. So I wonder if we're truly capturing all the injury that might be happening for these. These bioassays are set up for annual plants, not perennials. And maybe we're missing some of these results. One of the other things that I'm concerned about is now we're mixing multiple active ingredients. And so how is the injury associated? Is it additive or is it going to be synergistic and we're going to get more injury if we have different AIs that are present at different levels in the soil? So I think there's a lot of questions that are out there that we need to answer. And I'm fairly interested in this topic. But what can you do to minimize impacts? I think the number one thing you can do is follow those label restrictions, apply at the label rates, follow those plant back restrictions that are on that product, and look at those footnotes really carefully. We spend a lot of time and energy trying to summarize those results, and we put them in resources like A3646, which you can download for free at our PAT store, and I have the link below. In those, we have these really cool tables that you can see here that give you lots of details on when those plant back restrictions are for alfalfa. This will not replace reading the label, but it'll help get you a good head start. So really follow those plant back label restrictions as much as you can. I think if you can't follow those label restrictions, one of the things we recommend is plant a crop that you know is going to be tolerant. Also keep in mind the climatic conditions after you make an application in the next crop. In particular, pay attention if you have dry conditions, drought conditions. These 90+% of these herbicides are breaking down the degradation from microbes in the soil. When it's dry, we're getting less breakdown of that herbicide. So that's when we're going to have carryover issues when it's hot and dry conditions. So be on the lookout. If you do have a year like 2012, be on the lookout in the following year for herbicide carryover and plan for it. I think it's not a bad idea to rotate the herbicide mode of action to avoid buildup in the soil. If we're moving around our AI, that always can help because that can be an issue with some herbicides. And if you are concerned that you're maybe not at that level, doing some sort of tillage and working the soil can be a real advantage. And that does two things. One of the things it does when you work the soil is it actually is diluting the herbicide. Often the herbicide is in a fine layer where the plants can take that up and take it up in a concentrated value to cause injury. Working the soil will dilute it across that entire area, so it might be below that minimum threshold for damage. And what working the soil also does is it enhances microbial activity, and that might enhance breakdown as well. One final thing that we're looking at that we're interested in, and we're not sure if this is going to work, but we're wondering if planting cover crops might help enhance that breakdown as well, because those cover crops will be taking up herbicide. They'll be enhancing microbial activity as well. And that's one of the areas that we're working on. So with the small time I have left, I'll just summarize reminding you all that you probably already know this, alfalfa is an important crop in our rotation system, and we get lots of benefits from this crop. And so I'd encourage you to think about this as this crop as a solution to many of our problems. Changes in weed management practices and approaches have likely increased the precedence of herbicide carryover in establishing alfalfa. And so we just need to acknowledge these practices have changed, and we need to be more aware of this issue. And so really just take extra care when you're planning your crops the next year, that you're following those plant back restrictions, and be aware of any environmental conditions that might have led to enhanced carryover. And we're conducting some ongoing research, evaluating some of these other impacts and potential solutions. So stay posted. Feel free to communicate with me if you have other questions or ideas or concerns. With that, I'd like to thank you and highlight some outstanding people standing in an alfalfa field from many years ago. Just to highlight, the key is to get this information to others. There's my contact information with my email. You can find lots of this information on my website, as well as the NPM website. And with that, I'll love to stop sharing and pause and see if there are any questions that the group may have, if I have time.
Yeah, I know you got to head out in a little bit here, Mark. So if you got time for a question, there's one in the chat box. Is the increase in no-till farming another reason for increased injury to alfalfa?
Yeah, Brian, that's a good question. And I think that's likely another potential reason. So a couple things with no-till is, in no-till systems, we're more reliant on those residual herbicides, I would argue. And also we're not mixing up that soil, so it gives more of a chance too. So absolutely, that's another factor.
One came in directly, Mark. You got time for one more?
I have time until Steve kicks me off.
Okay, all right. I know you got another meeting to head to. Real quick one here that just came in directly is, so with the establishment, is there, after establishment, if the stand looks okay, but there's still some of those, a little residual out there between that 10 and 100,000 parts per billion, could that affect the longevity, even though the stand starts there? Can that lead to other stresses? Such as
and so I don't know the answer to that. I know that Rodrigo's done a little bit of work in soybeans and feels that there isn't a long-lasting effect. My big concern in alfalfa and the difference between soybeans is we're talking about a perennial versus an annual. So we have observed when we're looking at the roots that we're getting a different kind of root architecture development at our higher rates. Not getting as many fine roots and root hairs, not as much branching. And so that causes concern, but it hasn't been looked at to my knowledge. And I think it's a fair question to evaluate. There may be some data out there that I haven't seen, but that's a concern. But there's clearly going to be a level, right? And so I want to be clear that it's really going to depend on what that concentration is. And so I think it's worth spend a little bit more time trying to figure out where that threshold is.
That's good. Thank you, Mark. I don't see any other questions that have come in. So appreciate you taking the time, and if we have any questions, we'll reach out to you.
Great. Yeah, feel free to email me if any other questions. I'd be happy to answer it, or any other thoughts or observations. Take care, everyone.
All right. Thank you, Mark. All right, we're going to transition over to Steve Okonek. Steve is our agriculture educator in the Trempealeau, Jackson County area. And Steve's going to talk about some alfalfa pest management strategies. So Steve, looks like you're coming through. Slides look good. So.
Okay, thank you, Mark and Jerry. So yeah, talking about alfalfa insect management, and we're not advancing.
And we were working earlier. Maybe just try reloading it again, Steve. Sometimes that helps. Are you stuck? Okay, I try again, Steve.
Okay, are you seeing it now?
Yep, it's there. Are you trying to advance that lower left-hand corner of the slide?
Okay, now we are advancing.
There we go. Now it's working.
Okay, so we have two main insects that we're concerned about. There's several others that you'll see in alfalfa, but two main insects, that is the alfalfa weevil and the potato leafhopper. The alfalfa weevil tends to be an early season pest. The damage usually aligns with first crop harvest, and under heavy infestations, we can see a prevention of regrowth of the second crop after the first crop cutting. Potato leafhopper tends to be a mid-season pest, but potato leafhopper does the largest economic damage in Wisconsin and does not overwinter in Wisconsin, which has some unique challenges for us. On this slide, we have the alfalfa weevil larvae on the left and the adult on the right. Now, the larva stage is the most destructive stage, and we have different instars here on the left-hand side, or developmental stages of the larva. So they start out small, cream color white, they turn green as they ingest green material. And the feeding that we will see is this shot holing or pinholing of the leaves. It's not until we get heavy infestations that we actually get defoliation. The spring of 1989 on the western side of the state here, we had especially heavy weevil pressure. And then we had on late cut first crop fields, we had the top 6-8 inches of the field that were devoid of leaves, and the field had a gray cast. But that's about the heaviest I've seen. So they're not a pest that we're going to deal with every year. But the life cycle is as follows here. The adults emerge in the spring from fence rows or other undisturbed areas, probably waterways, maybe some buffer strips, that sort of thing. They lay eggs in the alfalfa field, and then the larva feeds on the tips of the alfalfa mid to late May. The larvae then spin cocoons and pupate to adults in early to mid-June. The adults will feed in alfalfa after emergence, but we seldom ever see any economic damage from adults. You will see a few adults here and there in your sweep net as you're checking for potato leafhoppers. The adults will lay a few eggs in the fall, but most of the egg laying that we're concerned about occurs in the spring. The adults then go into sheltered areas with mulch to overwinter in fence rows in those undisturbed areas. So the biggest thing, if you only take away one item from my presentation today, that is to scout. Okay, so we have a parasitic wasp that controls the alfalfa weevil. They lay eggs in the larvae, and that kills the larvae. If we're just out just spraying, recreational spraying, we're going to kill those parasitic wasps also with the insecticide. So make sure you scout, make sure you're at that 40% tip feeding. So that's 40% of the plants have pinhole feeding on the tips of the plants. It's not 40% defoliation. Okay, so the way you scout for alfalfa weevil larvae in first crop is you go around and make a W-shaped pattern in the field, pick 100 stems of alfalfa, the top 6 to 8 inches of the alfalfa plant, and then you look through those leaves looking for the pinhole feeding. So if you're 10 days or less from a planned harvest, go ahead and cut that crop. Most of our insecticides will have a pre-harvest interval that may actually delay your harvest past an optimum stage for dairy quality haylage. So if you're in that 10-day window, just go ahead and cut the crop, then monitor and scout the regrowth after harvest. So even if you've done your pre-harvest scouting for first crop, you're well below 40% or greater tip feeding, you still need to monitor that regrowth after first crop to make sure that we didn't have late emerging weevils or anything like that preventing regrowth. So what we're looking for on the regrowth of what will be our second cutting is 50% or more of the crown regrowth is prevented by weevil larva feeding. So those weevil larvae will sit there on that crown and they'll find a bud and they'll just sit there and they'll feed on that bud as fast as that bud is trying to regrow, and you will not get green up in a field. I've also seen fields where there's striping in the fields where the swaths laid. Those larvae were protected from bird feeding and sun and that sort of thing. And they're protected under there and they just live under that swath. Once the swaths are harvested, then we still have regrowth prevented in those areas. So watch those areas also. Use a labeled insecticide if you're at either one of these treatment levels. As Mark stated, publication A3646, Pest Management in Wisconsin Field Crops, has suggested insecticides. Also, some of the pre-harvest interval information is in there. That's not the label, okay? But use that as a reference. It's a good handy reference. Always read and follow the label, being especially aware for honeybee and that sort of information for protection of those. So that is the alfalfa weevil larva, alfalfa weevil. Our next and most destructive pest is a potato leafhopper. Okay, so the adults are an 1/8 inch long. You can see that in the picture there, that the adults have wings. They do fly short distances. The nymphs are wingless. They are very small when they first hatch out. So as you're scouting for those with a sweep net is the only way to scout for potato leafhoppers. You know, look close for the nymphs, and nymphs and adults all count the same as you're counting leafhoppers in your sweep net. Leafhoppers do not overwinter in Wisconsin and the upper Midwest. They reside down in the Gulf states, and they blow up on storms and other weather patterns that come from the south. So if we get those storms that come straight up from the southwest, that's going to be a good leafhopper year. If we get our precipitation coming mainly from the west, we will still have leafhoppers, but generally at lower levels here in Wisconsin. But scouting is required. There's no natural predators or diseases of any consequence that affect leafhoppers. So once they show up, they tend to multiply and live quite well in our environment. Now, the way they feed is unique because you will not see a defoliation. They have a piercing mouth, kind of like a mosquito has, and they feed on the sap in the plants, and they inject a toxin as they feed. That toxin breaks down cell walls And so then they're able to suck the sap out of the plant. The toxin is translocated throughout the plant and it stunts the growth of the plant. Economic damage occurs before visual symptoms. So if people are relying on visual symptoms, they've missed the boat, okay? Here is an example of visual symptoms of potato leafhopper feeding. It's yellowed leaves, the feeding is typically a V shape on the leaf. The leafhoppers will feed on the bottom side of the leaf, so they're sheltered from the sun and rain and that sort of thing. And they'll stick their mouth part into the bottom of the leaf and start sucking sap out, and they will kill the leaf from their point of injection out to the end of the leaf. And then they'll move back on the leaf and do it again, and then move to a new leaf. And eventually, if the field, particularly under warm, dry conditions, and especially on sandier soils, the field will get a yellow cast, and in extreme cases, it can even get kind of a red cast to it. A lot of people will say, well, it's dry, that's why our field is turning yellow. Typically, alfalfa in Wisconsin does not turn yellow from drought here, so it is leafhopper burn. In that case, harvest the field, scout, sweep again and see what the leafhopper numbers are and spray the regrowth. So to scout for the leafhoppers, you need a 15-inch sweep net, sweep in a pendulum motion in front of you as you walk across the field. There's YouTube videos out there of how to sweep for leafhoppers, so we don't need to go into great detail. We're limited on time here. Do 5 sets of 20 sweeps, so 100 sweeps. Count the adults and the nymphs. Keep a running total of your hopper counts, and then divide by 100 when you're done with your 5 sets. Okay. The threshold is based on the height of the alfalfa. Now, under heavy numbers, you can easily find potato leafhoppers between swaths as a chopper's going through the field in bare stubble. But typically you want to wait till you have an inch or two of regrowth there before you're out sweeping. So in the 0 to 3 inch height, 0.2 leafhoppers per sweep. So per swing of the net, you want 0.2 leafhoppers, so low numbers. 6 inch height, half a leafhopper. 8 to 11 inch, 1 leafhopper. 12 to 14 inch, there is a threshold. I kind of question why you would spray 12 to 14 inch height alfalfa. You've probably already sustained damage and you're probably close to harvest anyway, but then you need 2 leafhoppers per sweep. Typically wait 2 to 3 days after a heavy rain to sweep for leafhoppers, especially on real small regrowth or bare stubble. The rain will knock them down into the mud, but they always seem to crawl back out and come back. Numbers typically drop in mid-August. Sorry, wrong A month. Numbers typically drop in mid-August. So, don't be surprised if you're out in a field that had some damage on that second or third cutting, and then you're not seeing much for numbers, because that typically happens. So use a labeled insecticide. Always read and follow labels. Okay, but the leafhopper is the most destructive pest we have out there. Now on new seedings, especially if you're using a small grain and taking that small grain crop all the way to grain, we will have heavy damage underneath that small grain. That's one potential drawback of using a nurse crop. Now, if we're using a small grain or a small grain mix for forage, we're typically taking that forage off, mid-June or so, late June, then come back in and be sure to check that new seeding, because those new seedings are especially vulnerable to potato leafhoppers. And the damage that potato leafhoppers do to a crop, whether it's a new seeding year or subsequent production years, will affect the ability of that alfalfa plant to overwinter. So we want to make sure that we're controlling these things. But please scout, because the cut harvest spray option, we can do some damage, not only to the beneficial insects that are out there, but also to our pollinators, our honeybees and native pollinators. So let's not just be throwing insecticides out there without knowing that we need them. So scout, scout, scout. We do have some other insect pests out there: blister beetles, plant bugs, aphids, grasshoppers. Alfalfa blotch leaf miner is a unique insect because most of our insects either move south to north here in the United States or west to east. This little rascal actually moved from the east in Ontario to the west into Wisconsin, late 90s, early 2000s. But that is also being controlled by a beneficial insect. So we have these other insects out there that can feed in alfalfa and can cause some issues for us, particularly blister beetles and hay that might be targeted towards horses. So we do want to watch for these, but we seldom see threshold levels of these insects, especially just on their own. As we're spraying for potato leafhoppers or alfalfa weevils, we will control these other insects also. So that might be why they're not, reaching threshold levels. But publication A3646 has thresholds for all of these other insects. And so use that publication and it will help you as you go through the season scouting alfalfa. So any questions?
Well, thank you, Steve. I think we got one question regarding the drop in the potato leafhopper population in August. Is that due to time or more of a weather situation, or what's the reason?
I don't know. To answer that with evidence-based information, I don't have that information. I don't know what it is. If it's our day length is shortening, our nights are cooling, I'm not sure what it is, but it does happen. So
thanks. All right, thanks, Steve. We're gonna call up Mike Bertram next for our next speaker. Mike is the superintendent at the Arlington Research Station. He's going to discuss the Wisconsin Alfalfa Yield and Persistent Project, give us an update on that project and where it's headed. And Mike, it looks like things are coming through. Just got to put it in slide mode, I guess.
All right, and I'm unmuted, so can you hear me okay?
Yep, you looks good, Mike.
All right, well, thanks, Jerry. I'm going to talk about a study we've been doing for quite a while here in Wisconsin. It came about, I think, in the winter of 2007. Our extension Forage team was meeting and realizing Dan Understander was getting these 8 or 10 ton yields on small plots, but we really didn't know what producers were getting in their fields. So we came up with this protocol, decided let's try to just characterize what is actually going on in fields. And to do that, we had a couple objectives: verify the yield and quality of the alfalfa harvested over the life of a stand. So we called the life of a stand starting with the first production year being the year after seeding, and then going as many years as it was in before it was either killed off or tore up. And then second objective was to quantify how the stand, especially yield, changes over the life of the stand. And then an objective that came up in the last few years as we built this data setup was to provide this data to other researchers that were seeking to answer the questions about alfalfa growth, yield, and quality. And one of those was Valentin Picasso and one of his grad students who were following up, doing like an alfalfa yield gap study, looking at some of the factors that went into producing some of these high yield fields. So we worked with extension educators in their counties through Forage councils, primarily to identify the producers that were willing to cooperate. And we wanted producers with a vested interest because it is a multi-year commitment. I mean, we both wanted to have something to gain from it. We'd identify the potential fields, verify the exact acreage, and then we wanted to stress this isn't a yield contest. We just want no special management, whatever producers are naturally doing. And which would mean what, no special varieties, whatever they're going to seed, whatever they're going to spray, whenever they're going to harvest, and whenever they're going to rotate out. And we collected the data by weighing all the forage at each cutting. And this was aided by a lot of farms having drive over truck scales now. So many were doing that already. Sampling forage from fields at each cutting, and we take 2 samples per field so we can average them. A lot of farms were doing that as well. And then putting all the data together at the end and coming up with a report each year. So just a background on where we've been in 16 years. We've monitored 128 fields, and that amounts to about 308 site years because most are harvested 2 to 3 years, some 4. The majority of the fields are spring seeded. 107 versus 21 were late summer seeded, and but regardless, we don't count that first production year until that year after seeding. Seeding rates were typical, 12 to 28, most in the high teens. Field sizes, we weren't picky, as small as 5 acres up to 285 acres. The average was about 61. And then whatever the farm used for a cut schedule, 4 was the most common. Some more north had 3, some had 3 in a fall cut, some had 4 in a fall cut, and then even some years like 2012, we had quite a few fields with 5 cuts. And then in more recent years, some of the varieties had reduced lignin. This is where the fields were through the state. Yellow colored counties are where fields have been at any time during the 16 years. The counties with stars are current or 2022 fields. The star doesn't signify right where the farm is. We don't publish the farm's name at all with this, but I can disclose the Columbia County ones were at the Arlington Research Station. I generally put a new field in each year and then track that. So there's always a couple fields here being monitored. We had a pretty good response in 2021 with 12 fields. We still have a couple from 2020 left. 2022 is a little slow. Just the one from Arlington was the only one, but I'll put out an ask later for anyone who's interested. We'd like to get those numbers back up in 23. And you can see we generally had about 20 to 25 per year. And over the life of this, we've monitored about 7,800 acres and collected over 72,000 ton of dry matter. It was about 900 acres and 3,500 ton this past year. This is the form we use. It's pretty simple for collecting data. The only thing we really need from the producers are the cutting and harvest dates and the total tons that come off. We get dry matter and quality from the samples that go through the lab. And we do the rest of the calculations with the sheet. And we try to share this back with the producer at the end of the season. So they're getting some numbers back as well. So getting into some of the data, this is the average date of each cut over the 16 years. And it actually maybe isn't that surprising. The average was 30th of May, the 30th of June, the 30th of July, and the 29th of August. So pretty much right at the end of each month. It has ranged from as early as the 16th of May in 2012 to the 10th of June the next year for first cut. But regardless of when that first cut is, a lot of the fourth cuts are right at the end of August. In years where there was a fifth cut on some fields, it usually was by Labor Day. And the average timing of a fall cut when there were fall cuts was in October 12th. And in 2022, we were really no different than the average or the last couple of years on first cut, and the later cuts were about 3 to 4 days earlier than what we had seen over the long-term average. Moving into dry matter, we kind of saw each cut, and for season average historically, and I guess I'll explain this chart just so everyone can see that. The bars are showing the high, the low, and the mean for each, and then the diamond is what it was in 2022. So in through the mean through the long term has been about 45, ranging from 44 to 46, and then 45 for a full season. And this is a little drier than we'd seen for recommendations for storage, but it doesn't seem to be causing a problem. And that would be 55% or so moisture. In 2022, we were pretty close to the long-term normal for the first three cuts. A little less dry matter in fourth cut, but I think we had a little wetter weather in August that forage was made a little bit, went up a little bit more moisture in it. Then the one everyone cares about is the yield. And this shows yield by cut and same type of format. So like on first cut, we normally see about 1.5 tons, 1.56 of dry matter yield. It's ranged from a low year of 1.22 to a high year of 1.84. We were pretty close, just slightly above in 2022. Where we really got hit in 2022 was second cut. Normally about 1.1, we were only at 0.8, and that was the lowest second cut yield we've seen of the 16 years. Third cut was just slightly below normal, and fourth cut was actually the second best yield we've seen. And not exactly sure why. I know a second cut tends to sometimes be a little closer in timing, especially if first cut was later. The average was only 26 days of the 15 fields. So that might have affected that yield. I think weather also plays a role in that as well. Was on the 4 cut fields, it was 28 days to third cut, and then actually 33 days to fourth cut, and that might be why fourth cut had a little better yield. And then this has season yields. So start off with 2022, 4.1 ton was our average for the year of all the 15 fields. It was similar. It was actually about a 1/4 ton less than the long-term average of 4.36. And it was actually the 5th lowest of the 16 years, similar to 2017 and 2011. The best years we've had were 2007 with just over 5 ton, as well as 2010. And then 2021, we had about 4.9 ton. The lowest years were 2020 with 3.6 and 3.7 in 2013. The other thing, I guess, is we had kind of a compressed range where 4.8 was the best field in 2022, and that was the lowest of all these best fields each year. And the 3.16 was actually one of the higher of the low fields. So compared to some of the big ranges in some years, we just didn't have that in 2022. And that shows in this chart, just we didn't have anything below 3, which has kind of been the lowest yields, and anything above even 5 ton. Generally 6 ton is the highest fields we see. And then this is the distribution through the 16 years, and it shows a nice curve, pretty good distribution. The mean was 4.36, so it's clustered more around that. I mentioned our best fields were just above 6, 13 times we had that. 22 times we were below 3, and actually a lot of those were since 2019, because 2020 was the lowest year. I mentioned at 3.6, and we actually had 10 of these 22 fields were in the last couple of years. And a lot of that's attributed to the winter kill we saw in Northeast Wisconsin. In some cases, the producers had to keep the fields in just because they needed forage, even though it wasn't a great yield, it was something. Up until a couple of years ago, we just focused on the top, or everything was focused on annual yields, but I was getting a lot of questions like, what are the yields over the life of the stand? So it compiled some numbers. The most common was 3 seasons, and we had 44 fields that fell into this. And this is fields that are done and current fields. But we averaged 13.4 essentially tons, which is kind of right at that 4.4 to 4.5 ton to the acre. The best field that was in for 3 seasons had 18 ton. The lowest was 9 ton. The highest yielding ones come after 4 seasons. 16 of those fields, we had one that almost got to 22 tons over 4 years. The lowest was closer to 12. The mean was about 18. And through that mean, it's roughly 4 to 4.5 ton per year. Not much different than what we see for the total. And then it was also kind of asked, like, what are the top fields? And this a lot of information here, but it shows by the number of seasons that a field was cut, what the top producing fields were. And about 16 fields were made it through 4 seasons after the production year. And the best one was 21.8. We had 5 that were 21.4 and above. Lower in the chart, it's kind of dominated by 3 fields, 114, 111, and 618. And it's the year is 14, and then like the first field of that year. So they're all from Fond du Lac County. They actually were all from the same farm. So that producer has something figured out, because over 3 years, he has the highest 3 yields. And those fields kind of dominate when you look at a combination of 2 best years, if it was the first and second, or in one case, the second and third years. And they kind of dominate the single best year, either happening the first year or the second year in the cut schedule. However, the best 1-year yield we had was actually last year in Outagamie County, 6.6 ton of dry matter over that of that year. And that field had a fall cut, which the fall cut added enough yield to actually put that in the first place. So that was kind of the first goal, yield. The next was looking at how yields change. And we did this in 2 parts. One was looking at through the season, and then the other was over seasons. And this gives an example of how the distribution of the yield per cut. So if you're cutting 3 times, which was 43 of our site years, you get almost half your yield in the first cut and approximately a quarter in each of the other cuts. The majority were cut 4 times. You get just over a third in the first cut, about a quarter in the second, and about a fifth or so in the later cuts. And if you cut 5 times, you get about a third in the first cut, and then it just keeps going down to later cuts. And then over years, so we compared all this to the first production year. And so the second production year, was 101%. So basically about the same as the first production year. The second year, we dropped off about 5% from that first year, which is still really good. And then down to 81% by the fourth production year. So, and that's a fields, a lot of fields didn't make it to that fourth production year. So those are the fields that producers decided were good enough to harvest and keep harvesting. So it's basically prudent to keep a stand about 3 years after that first production year. And after that, you're generally going to see yields starting to go down, but each field really has to be judged independently with looking at yields coming off of it and stand counts to make those decisions. I guess in the interest of time, I've got a ton of quality charts, but just to show, I don't have time to look at all to show all that data, but overall quality parameters, and these charts work about the same. Crude protein, we normally see about 21.6%. We were 22.5% this year, which was the second highest we saw after 2020. And generally what we're seeing is when we have good yield years, the quality is lower. When we have lower yield years, the quality is better. So most of our better quality years were 2013 and 2020. NDF was pretty close to normal. NDF digestibility was the second highest we've seen through the 16 years, as was RFQ. The average relative forage quality was 200, weighted with all the cuts and total season. And then milk per ton was actually a record in 2022. It's the previous we had seen was 33.12, and we surpassed that. So really good quality in 2022, even though we didn't have quite as much yield. Okay. So summary for this for 2022. We dropped 5 fields, mainly because the producers plan to rotate them out. We gained 1 new field, so we had 15. We had a near normal first cut date, and then earlier than normal dates in subsequent cuts. Season dry matter was right at what we normally saw through the study at that 45%. Yields were not as great as we've seen. It was the 5th lowest season dry matter yield of 16 years. Second cut was the lowest cut we had ever seen, and it was about 1/4 of a ton less than the average. But we did have good quality, the second highest protein digestibility in RFQ, and the most milk per ton that we've seen. And through the long term, our second and third production yields were nearly equal to the first production year, but then yield dropped off about 19% in that fourth production year. And to date, the average yield was 4.36, and almost 3/4 of the fields have yielded less than 5 ton of dry matter to the acre. So disappointing in that regard, especially when we know that the genetics is capable of reaching almost double yields of that. But we also know that on small plots, we don't put a lot of wheel traffic on, and there is a lot of wheel traffic generally on production fields trying to get those harvested. I want to acknowledge everyone who's helped on this through the years, starting with the county or extension educator coordinators. This year, especially Erica, Kevin, Tina, and Scott for helping in their counties with that. And then to all the producers who had to take a little bit extra time, maybe a little more, to grab a sample, share some of their data. And then Midwest Forage Association, since 2009, has been funding the forage analysis cost for this study, which is the major cost. I also have to recognize Mike Rankin, former Fond du Lac County educator. He was very instrumental in starting this, and he was the keeper of the data for the first 8 years. I've now actually had it for 8 years since he retired, so we're even on that regard. But he did a lot of the legwork to get this going. And finally, I mentioned we're down a little on fields. We'd love to have more into this for next year. So you don't have to be necessarily a extension educator to help with this. If there are crop consultants or anyone else who, especially if you're working with a client who's doing that, a lot of this already, it's not a lot of extra work to help share some of this data. We'd love to be in more parts of the state than just kind of the south to northeast corridor. So get a hold of me if you think you might be interested. We also could work straight with producers who might be willing to cooperate as well. So with that, Jerry, I think I've gone through the presentation, so I take questions if there's time.
Yeah, thanks, Mike. We do have a quick poll for folks to answer if they wouldn't mind. I think we've just got a couple questions here. So if people want to take some time to answer those questions while we're transitioning over to our next speaker. Just real quick, Mike, is there a plan? How many more years do you think they're going to do this project?
I wish I knew.
Okay, I had heard maybe they're trying to get 20 years of data because it's fun. So
yeah,
through 2027, so there would be time yet for producers to get involved.
we don't have a plan to retire it yet, but I know it is one of MFA's high-ranking priorities for funding, and it's not a lot of cost to run the samples either. So it's a fairly low cost study or survey to keep track.
Sure. There is a question in the chat. If you want to answer that for the sake of time, Mike, we're just going to move on to John Jones. But if you want to answer the question, excuse me, it's directly to me. I'm going to have to ask you this one. Do you know why the farm in Fond du Lac was consistently high yielding?
I actually don't, because I don't work directly with the farmer or the producer. It's the county educator that does more of that direct. I do know who it is, but I'm not sure, but I do know some of that was the idea of the follow-up with the yield gap study to try to survey some of these producers to find out exactly what they're doing, to see if common themes emerge for success with growing alfalfa.
Okay. Yeah, well, thanks, Mike. Appreciate the information. That's always good stuff that helps us with recommendations and working directly with producers. So appreciate the effort and keeping that going for us. All right, we're going to transition over to our final speaker, Dr. John Jones. He's a UW-Madison soil science researcher. John's going to talk to us today about monitoring sulfur in alfalfa and fertilizer source comparisons. So John, I think if you want to share your slides, I think we should be good. You're coming through so far.
Yep. Can you see that all right, Jerry?
Yeah, looks good and you sound good. So I think you're all set, John.
All right, we'll be good to go. Well, thanks for the opportunity to come speak today. I appreciate kind of being able to round out the discussion today. We've had some really important topics on alfalfa production and maybe what some of the limitations are. One of the topics that I was asked to talk about and that have some interest in pursuing to a greater degree in the future are monitoring sulfur nutrition within alfalfa fields. Again, we're tying down on alfalfa now, but I think it is certainly a question that we have across many crops in the state of Wisconsin. But today I'll be talking about some trends in soil and tissue analyses and looking at those relationships and really the fertilizer source considerations for what options you have in terms of sulfur fertility and nutrition, because there are a lot and they don't all work in the same environments to the same degree. Let's see. There we go. All right, so quick presentation overview, three bullets. So what I want to hit on, I kind of swapped the order of the presentation because I think it makes sense logically. So I'll overview sulfur fertilizer sources and what's available, and how some of them compare in terms of their placement and composition. Then I'll talk through some results of soil and tissue analysis, some sulfur response studies that do kind of look at some nutrient interactions that have been done throughout the state. And then I'll talk about a little, some preliminary data on a alfalfa fertility survey, specifically on the sulfur numbers. And then I'll share a few slides from a colleague from Minnesota who has some really nice sulfur source response studies. So challenges across the state of Wisconsin that we have for sulfur management, but also many other nutrients as well. You have a map of soil organic matter percentages on the left and sand percentages on the right. Why are those important? Well, they really interact when we talk about soil moisture status, soil temperature, which you'll see is really important in terms of sulfur fertilization and sulfur availability. And one of the things that I really think maybe underappreciated about Wisconsin soils are that even if you're in an area working with a silt loam soil, sometimes because of the influence of soil parent material in Wisconsin and honestly, sometimes the impact of the central sands have had throughout the last few centuries, that we do have some silt loams that are maybe a little higher percent sand than what you'd find in Illinois or other parts of the Midwest. And I really think that influences a lot of the nutrient management that we see across our state, and where maybe some soils are underrepresented when we talk about recommendations and guidance for fertilization. So, but in the context of sulfur, a lot of times we link soil organic matter to sulfur recommendations. I know some states directly use soil organic matter, but certainly all of these really play into where a specific product can work for your alfalfa crop. So this wouldn't be a sulfur presentation if I didn't talk about reductions in atmospheric deposition of sulfur. I think that's kind of a expected slide when we're talking about this. So on the left is a figure that Keith Kelling and Emmett Schulte put together in the early 90s, partitioning out two separate areas in Wisconsin where there is assumed to be a certain amount of sulfur delivered via precipitation, and in either 10 or 20 pounds, depending on essentially how close you are to Chicago and the Rust Belt, really, in the United States. Jump forward to a summary about 2 or 3 years ago now that was done by the atmospheric deposition program across the US, and has different monitoring sites and models, different types of deposition of all nutrients and constituents in precipitation. But you can see that Wisconsin is fairly low, and so is most of the US. You can see a few hot spots that may be due to manufacturing or simple sediment transport or aeolian transport of sediment with wind. But in general, most of the state of Wisconsin is in that 4.5 pound per acre per year of wet deposition of sulfate specifically. And it's important to remember that the map on the left is sulfate sulfur. So if you convert that, the map on the right is even lower. So it's something to consider, is that we don't have that atmospheric source of sulfur like we did. A simple sulfur cycle or sulfur cycle diagram to consider what are the points in the soil and plant schematic that you're looking at we're talking about today. Jerry, can you see my my cursor?
Yep, we can see it.
Okay, perfect. That's great. And so really what there we go. What we're going to be talking about and hitting on today is where when sulfur sources are applied, this oxidation process that has to occur if an elemental sulfur source is applied to convert microbially to sulfate, which is then taken up by the plant, because we do require sulfur in the sulfate form to be taken up into our crops. And so obviously we have all these different inputs and outputs of sulfur within our cropping systems, but today we're really going to be hitting on kind of this elemental sulfur conversion and sulfate uptake. So now on to kind of the one of the biggest chunks of the talk, or what information I'd like to share is what I kind of just described. Sulfur is taken up in the sulfate form. Within fertilizers, we do have sulfates, elemental sulfur. Thiosulfates are another kind of more complex sulfur molecule that needs to be converted to sulfate to be taken up. But these kind of three main sources are what I think mostly you'll see out at the farm gate and maybe with your ag retailer. And so if you are applying a product with elemental sulfur or elemental sulfur itself in the those small yellow pellets that you'll see, that does have to be oxidized, and that's driven by microbial activity. But really, when you boil it down, it's temperature, moisture, but then also the particle size of that fertilizer, and specifically that sulfur, can really play a big role. We'll get into that a little bit more. Mineralization of organic sulfur is something that we certainly can consider, and why we do look at sulfur management probably differently in our sandy versus loamy soils with maybe a higher organic matter percentage as well. So again, I'll talk about sulfur oxidation at a few points, and you'll hear me reference it, but I just want to review. This is the process that I'm talking about. So this elemental sulfur in the yellow circle on the left is getting converted to sulfate. Essentially, we need the right environment of water, oxygen, and then that promotes a certain microbial activity. And throughout this process, though, we do have acid production when you see those two H+ ions right there. So certainly when you look at if you look at the small figure on the right hand of your screen, you can see a relationship between soil pH and elemental sulfur oxidation. There's some great research that's come out of Australia in collaboration with some fertilizer companies that's done a lot of work into identifying how these relationships occur and exist within soil. Now, when you look at the optimum temperature, and we're talking about just chemically of this reaction, it's about 90 degrees Fahrenheit. Well, how often do we get to that environment when we really need sulfur uptake in Wisconsin soils and systems? That's another something to consider when we talk about sulfur sources. And if you are in a high pH, more alkaline soil system, it's not just that you're producing acid with this process, but in general, because of the way the reaction gets pushed at a higher pH, you can drive sulfur oxidation a little bit more. That's specifically what the Australians do look at quite a bit. Now, particle size, something that really is important, and it's kind of, if you talk about 4R, we talk about source rate, timing, and placement of nutrient stewardship. Particle size might be, I don't know how you'd fit it into the R, but maybe another consideration that's really important for sulfur. So if you can see this figure on the right here, again, out of an Australian group that looked at different particle sizes, what's probably really important is to look at those blue points or line is about 38 to 75 micrometers or microns in diameter. And you can see as we got bigger to the yellow points and the green points in terms of that diameter, the oxidation percentage was reduced in just a short incubation over time. And that really does reflect what's happening in our soils as well. So certainly when we get to smaller particle sizes, and that's always sometimes kind of hard to find and interpret that information, but most companies are pretty good about communicating what that is on the tech sheet or something for a given product. What's also really important is the dispersion of those particles once water temperature and our microbes start acting upon them. We can kind of think as a grouping of sulfur molecules as something that needs to be interacted from the surface out. I apologize if you hear some construction trucks beeping up. We're in eternal construction right now behind King Hall in Madison. But to get back to the point, dispersion, and we'll get back to that in the next slide, is really important within that soil system. One important point is that elemental sulfur is hydrophobic, and so it doesn't dissolve in water the same way a sulfate molecule like potassium or ammonium sulfate would. And so I mentioned dispersion of elemental sulfur, and here's again some really nice figures from that same Australian group that's looking at essentially when you move from left to right, you have, I apologize for not the labels aren't showing up. But you have elemental sulfur in the A panel on your left. In the center, you have essentially a sulfate product that is not purely pure elemental sulfur. And then on the right, you have a co-granulated mixture of monoammonium phosphate and elemental sulfur. And you can see if we have that kind of very concentrated granule that essentially is made up of mostly elemental sulfur, we see that dispersion really be inhibited. And that's probably the case of a few different interactions between temperature and moisture throughout the soil system. Again, this is done in isolation here. But you can see that over time, and again, you're moving from time 1 on the upper panels to time 2 on the bottom panels. You do see a pretty big difference in products, and so that's really important. On the far right, you can see where that monoammonium phosphate is still left over, although the sulfur components have started to break down. And the figure on the right here with the kind of the orange particle and then essentially the yellow sulfur particles, or I should say the orange granular and the sulfur particles in the yellow, we can see that there's this surface effect. So we want the microbes and the moisture and the temperature to be able to interact with all surface layers of those sulfur particles. That can't happen if they're too bunched together and not dispersed enough. So that's why I talk about dispersion as being so important. Common sulfur products and some of their properties that we might consider within Wisconsin or the Midwest. There's a lot available, certainly more than I have listed up here. These are simple products that I'm familiar with and have worked with. But certainly we see gypsum used as a source, different sulfates, whether it be ammonium, potassium, depending on if you do want to look at it as an N or K source as well. Certainly if you're talking about a potassium sulfate product, that works well for a lot of forage systems. I've used that in some of my research when wanting to look at that interaction of K and S nutrition. What I also have on this table is the particle size on the right. And so you can see that when we start to get into elemental sulfur products, that sulfur particle size is something that's highlighted a little bit more. We have different thiosulfate products. That's mostly, from my experience, been used more in dissolved solutions in liquid formulation, specifically when you're starting to talk about fertigating crops. So we won't talk about that too much for alfalfa in Wisconsin. But when you get down to the products like elemental sulfur, Tiger 90, for example, the micro essentials 10 or 15% sulfur, and then MST, which is a nutrient product, certainly you can see that organic sulfur elemental sulfur is a constituent, sometimes a complete part of the sulfur source. In terms of the form, for micro essentials, for example, it's a 50-50 split of sorry, that shouldn't say organic, it should say elemental. Sometimes it's referred to as organic, but it's really just elemental sulfur. And then, for example, for the MST, you have elemental sulfur, but it's co-granulated with monoammonium phosphate, what I talked about before. Some of these have pretty similar. You can see the bolded and underlined sulfur contents. And remember, I talked about that kind of 30 to 50 to 60 microns was kind of that in the previous data, that ideal particle size for oxidation to occur for elemental sulfur sources. And you can see most of these products, for example, on the bottom 3 rows fit into that bin pretty well. We certainly do have acidification with sulfur sources, and I won't get into this figure too much. This is just a recent study and review that came out of some researchers that have a lot of experience in soil and fertilizer chemistry. But you can see that we do have acid produced in per pound of nutrient. You see this 3.313 number. In general, that's in kilograms per kilogram, but about 3 pounds of calcium carbonate is needed per 1 pound of sulfur that has to be oxidized. Remember, because two hydrogens are produced when that sulfur is oxidized. So we have to think about that. That'd be about 4.6 pounds of 65 neutralizing index ag lime is required per 1 pound of sulfur. And so certainly these are things to consider. Potassium and calcium sulfates are neutral. There's no pH effect similar to gypsum. But if elemental sulfur is a product that's being used quite a bit, monitoring soil pH with soil testing should be a good idea. One comment on intentional soil acidification, certainly that is something that gets talked about a lot. And I just wanted to show some data that was actually part of a soil pH study. Phil Holman is in charge of up at the Spooner Research Station. I've had the fortunate opportunity to work with him on. But essentially what we were doing with this study is trying to alter the soil pHs to get to some target ranges, because we're just looking at pH, not necessarily a liming or sulfur effect on crop growth. And this is working with a fairly coarse soil up in northwestern Wisconsin. But we applied elemental sulfur to reach target pH levels, again, 1 to 1.5 soil organic matter. And you can see the initial pHs, and then 12 months after that on the first and second columns moving from left to right. And certainly we did decrease the pH to a certain degree. If you look at rates that we applied are around 870 down to 350 pounds of sulfur per acre as elemental sulfur. And it is, in general, took about 760 down to 629 pounds of sulfur to drop 1 pH unit, right? If you just do the relationships between there. If you relate those to the recommendations within A2809 extension publication, this would be the pounds of sulfur per 1,000 foot to drop a pH unit. You look at that red squared number again. I pulled this this box out of the UW fertility recommendations document. And for the soils and 0.5 to 2% soil organic matter, we go down to a desired reduction in soil pH of 1 unit. And that's about 24. So, and granted, this is covering all soils. It's not soil specific. But essentially it makes sense that in a very coarse soil, we were able to be a little bit more efficient with that sulfur source at dropping the soil pH. So something to consider if that is something you're thinking about. But if you're getting into silt loam soils, high CECs, high clays, you need a lot of sulfur applied to move that pH. And then certainly manure as a sulfur source is something that needs to be considered. Testing pits, testing loads to get at least some values that might be able to guide whether if enough sulfur is being applied with those manure applications. Certainly when you look at the values in the red box on the right, we can have considerable amounts of S being delivered with manure source. So consider that in terms of fertilization guidelines. And so I'll kind of move to the next, sorry, we're catching up on time here, but I'm kind of move to the next part of the talk where I look into some results of some response studies. So I definitely understand if people need to not hop off, but I'll just kind of keep chugging away here. So potassium and sulfur effects on alfalfa yield. This was a study done by Carrie Lewosky and Todd Andrasky here in the soils department about 10 years ago now, or over 10 years ago now. But it was at 3 sites looking at Southwest, Northeast, and the North Central Wisconsin at Lancaster, Freedom, and Marshfield, looking at a plus or minus of added potassium and then different sulfur sources. And what's really interesting when you just look at the 3 sites that are shown on the right is we do have different trends in terms of which treatments we saw there was a response, or Carrie and Todd did see a response. So you look at the Freedom site, we actually see more of a potassium response than a sulfur response. You can see that the treatments on the 4 treatments on the right have added K. The 4 treatments on the left have no K and just sulfur added. Down at Lancaster, there was an interaction between potassium or excuse me, there was not an interaction. There was a sulfur response here, but in general, fairly low level of potassium response, only when there wasn't sulfur applied. So there probably was an interaction of plus or minus yes or no sulfur application, and then needed K as well. And then if you move down to the bottom right at the Marshfield site, essentially you're really just looking at a potassium response. There was really no change determining if there was 25 pounds of gypsum, 75 pounds of gypsum applied every year, or the elemental sulfur, the 75 pounds of S was only applied in the first year of production. So that's something to consider, that we have these really different responses at different sites. And let's see if this is. There we go. And when we just pull out those 2 sites, the one at Freedom and Marshfield, we have almost an exactly similar initial soil test K for these sites, but certainly a different interaction. At Freedom, we see a potassium and sulfur interaction because once we do get up to this level of K applied and the 75 pounds of gypsum annually, you do see an interaction compared to lower gypsum rates. But at Marshfield, it was really only a potassium effect. And so when we think about drainage conditions and properties of an Outagamie County soil compared to soil around Marshfield or around Wood County, essentially you can look at some pretty different, or Marathon County, excuse me, you can see some pretty different or consider some different drainage capacities, temperature to a certain extent, but this really makes a lot of sense that a lot of potassium and sulfur specifically is very site specific in its response. Optimum K is required for an S response in a lot of cases. This is one example of a study that Carrie did as well down at Lancaster on an alfalfa grass mix. This is just 1 year, but you can see it broken down into first, second, third cutting, and then the total yield for all three cuttings. And we can see that in general, we had an initial soil test K of 148 ppms, which should be considered optimum to high. But we saw that sulfur response that was indicative of what we would expect. Now, what's important here is when they're applying 25 pounds of elemental sulfur, just the S treatment there. So you have 25 pounds of gypsum, elemental sulfur, and then gypsum plus sulfur, so 50 pounds total. You really didn't see any benefit with the elemental sulfur. And this probably makes sense because a lot of times those elemental sulfur products, and this wasn't this was just a 100% sulfur elemental sulfur product. We really probably didn't have a very high oxidation rate. And so we did not have availability compared to sulfate that's readily available with gypsum. Potassium content can be affected by sulfur. This is just looking at different treatments and how that's interacted. Those are the three cuttings, first, second, and third. We don't need to get in that too much, just to try to get through this here. But certainly there also can be a potassium uptake and can be really affected by sulfur source as well. So this is that same grass alfalfa mixture study. If you look at the treatments that essentially have that added sulfur available, where 25 pounds of gypsum was applied, you're always increased in terms of your potassium uptake. So there's probably an interaction there that's really leading to probably more of a yield effect than a potassium concentration effect. But it certainly can affect the amount of K uptake. And if you're looking at your K balance and needed K nutrition further down the road, this can be really important. So think about these nutrients in tandem. And soil sulfur really isn't a very reliable metric in a lot of this prediction of a response. It's more of the environmental conditions that are existing. If you look at this, and this is data from the previous study I was discussing. I averaged across all the three sites, which is something that's not always the best to do because we want to take into consideration site specific variability. But just to look at how variable some of this was. The first timing on the far left, at least, so you're looking at 6-inch soil sulfur on the left and 3-foot soil sulfur in terms of pounds per acre on the right. Across the timing of the study or the 3 years of the study, there's no sulfur applied for that first date, but after that you have either an annual or first year application. And really we only saw differences when we had that 75 pounds per acre per year of gypsum applied. In terms of increasing soil sulfur, which we know is a very mobile nutrient within the soil. And so really when you pull this back to those yield data that we just looked at, these relationships don't really predict those yield responses. And so really when we talk about a soil test for sulfur, for example, probably not the most predictable and reliable metric of a sulfur need. This is some results, and I can kind of move through this quick from Dr. Dan Kaiser, colleague at University of Minnesota, that he had presented throughout the winter. Not getting into too much of the details on this, but essentially he was looking at different sulfur sources for alfalfa. He also has a similar project looking at corn response. But really when the sulfur variability in terms of a source effect wasn't really that high. When you look at those, that burgundy and yellow bars, you really didn't see a huge difference in terms of sulfate source or the MST product. And then you can see the green bar was Tiger 90 applied at 120 pounds of sulfur at pre-plant or pre-establishment. But certainly you did see a response over the control across these 2 years, and this would be over in Rosemont in a lust-derived deep lust soil. And essentially what Dan really found out was that a minimum of 10 pounds of sulfur was profitable here. And it led to about $160 per acre return when we think of $150 per ton toward forage cost or forage prices, and then about a 50 cent per pound sulfur price as well. I'll give some links later, but his blog on that study is a great resource. And so this is your UW recommendations for sulfur that you'll find in the whole recommendation book. It's just one little table in the page. So why is it so simplistic? Well, some of the data I think I've shown today is that we don't have that incremental rate response in terms of alfalfa and other crops sulfur additions. We're kind of at the point, and maybe this needs to be looked at a little bit more, but we're kind of at the point where there's a minimum threshold of sulfur fertility and nutrition we need. Let's meet that. And then above that, we're worrying about other yield or quality limiting factors. And so that's why when we look at, for example, forage legumes, you have higher rate and incorporated if it's incorporated at seeding. If it's top dressed, you're essentially looking at a lower rate, probably because of that oxidation and dispersion. One of the things that can reduce the dispersion of elemental sulfur, specifically in the soil, is when it is incorporated, you do get kind of a coating effect within soil colloid. So that's something important to think about as well. If you're looking at an organic sulfur source that needs oxidation, and think about how that interacts if you're top dressing that onto an alfalfa stand, that's a little bit different than if you're applying a sulfur source that's going to be available right away. So think about that timing aspect as well. So why is it so simplistic? Well, the data so far has shown that this system can work, but I will admit that in terms of responses to sulfur rates, a lot of many higher rates than what are in this table I've seen work fairly well and are needed in certain systems. So maybe something that we need to be looking into a little bit more. A quick overview, and I know I'm about 10 minutes over on time, is an alfalfa fertility study I'm working on. I'm now in the second year, had about 40 sites in 2022, looking to have around 50 in 2023. It's essentially a study where myself or someone from my team comes on. We take soil samples, tissue analysis, and plant samples, and are kind of mapping how nutrient what the either deficiencies or sufficiencies are existing across the state of Wisconsin. I'm also doing a little bit of work looking at some soil K testing methods, looking at mineralogy and exchangeable potassium. But if you're interested, this is a project that I'm continuing on this year as well. And I think the data that's coming back will be pretty cool to look at. Just some quick data that we're looking at. We see a nice relationship between soil test K and leaf tissue K. Sulfur, not so much, not surprising, but we're also seeing a nice relationship with boron that I'll get into a little bit more as the study progresses. And you can see the map of where the sites are so far in 2022. One thing to note is that when we do look at leaf tissue sulfur, so essentially you can see that 0.25 threshold that's commonly considered the lower end of our sufficiency range. We do have quite a few samples that came back lower than that. So something to think about going forward, and this will all be communicated once we get through the study a little bit more. Key takeaways. There's an interaction between your sulfur source and the system you're working in, whether that's in your year of establishment when you're applying it, or if you're top dressing sulfur. Think about how that product works in terms of its ability to oxidize if you're looking at using an elemental sulfur source. You can contact me. I've got a few more resources to look at how some of these products have fit into certain systems with other crops as well. Tissue analysis is probably one of the better tools to look at a minimum threshold of sulfur nutrition. Soil samples just not as reliable because of the mobility of sulfur within soil systems. So here's some resources. I thank you for your attention today. Sorry I went over a little bit, but an exciting topic, and I think there'll be some more research coming down the line on it.
Well, thank you,
Looks
John.
like we only lost 5 participants going 10 minutes over. So thank you everyone for staying on. I appreciate it.
Yeah, we got a riveted group today, so appreciate that. John, there is a question in the chat box
Okay.
And so if you want to answer that one based on the slide, would gypsum or elemental sulfur become available to a crop sooner? So if you want to.
Okay, so gypsum or elemental sulfur. So if you're working with an elemental sulfur product that's that kind of 90 to 100% sulfur, if you think of those kind of yellow pallets that the products that you're using, the sulfate and gypsum would become available sooner because sulfate is readily available. If you have rapid oxidation, that occurs soon enough. If you get that elemental sulfur on early enough before the crop's being grown as well, then you can allow for that oxidation to occur. I get a little worried about fall applications of elemental sulfur before the growing season, only because usually that's going to be in tandem with some phosphorus as well, and so we want to try to keep that trying to reduce some P losses in the fall. But and the sulfate and gypsum can be available a lot sooner than elemental sulfur can within that 90 to 100% sulfur product. Now, I will say if you're looking at The micro essentials products or the MST products that have a lower percent elemental sulfur that's either co-granulated with MAP or some phosphorus sources. Because the sulfur particles are dispersed in the granule a little more evenly and spread apart. If you compare that to an elemental sulfur particle, that can oxidize a little bit faster. So that is something to consider. So, in terms of ranking them, that's a little hard to do because they interact with the environment differently. But if you're just talking about that 90-100% sulfur, I would want to, if you want it quickly, I would look at a sulfate sulfur source.
Checking the chat box. I didn't see any more questions for you, John. So I think we'll wrap things up. Again, we'll